by dihybrid? What is meant by dihybrid? So when we made a cross between two parental plants which exhibit or differ in two traits. సంకరణంలో ఉపయోగించే రెండు జనక మొక్కలు పరస్పర వ్యతిరేక లక్షణాలు కలిగి ఉండాలి. ఎన్ని జతల పరస్పర వ్యతిరేక లక్షణాలు కలిగి ఉండాలి? రెండు జతల. డైహైబ్రిడ్ అంటే we can consider at a time two characters. What are the two characters here? That is, just we can go for once minute. What are the round? Round is indicates what? Shape of the seed. Yellow is what? Color of the seed. Yes, and at a time we can consider two traits. Two opposite contrast characters are observed in the parents at a time. Here round opposite contrast character is what? Wrinkled.
Another good evening.
Hello, any problem?
Sir, if possible, please erase this monohybrid cross, capital T, small t, and capital N, capital N.
Then right? Is it
Okay,
clear now?
Proceed, sir.
Okay. So these at a time with two characters. At a time we consider two characters which are differ in the parental. That is shape of the seed and color of the seed, and having two contrast characters. So that you can select two parental plants. They are homozygous and also differ in two pairs of characters. So in the F1 hybrid, we get all are heterozygous and a dominant character. According to this, round is the dominant character and yellow is the dominant character. Which one is missing? Wrinkled and green. There is no chance of wrinkle and green character in the F1 generation. Why? Because wrinkle and green representatives are in the small letters, and round and yellow are the dominant one, which are expressed in, which are denoted in the capital letters. Capital letters is nothing but F1 only. Plants shows such type of character only. And we can go for F2. The ratio is 9:3:3:1. So this dihybrid experiments explain about law of independent assortment. Law of independent assortment. Next we can go for next slide. So this is the table Punnett square with the dihybrid cross. So here we are getting 9:3:3:1 ratio in the F2 generation. That is phenotypic ratio, and genotypic ratio is nothing but 4 2s and 4 1s. So we can easily remember the genotypic ratio for dihybrid. We can write first 4. Okay, 2 write 4 times. The total progeny is 16. The total progeny is 4 2s, 4 1s. So 4 2s, 4 1s. Like that, we are getting total progeny in the F2 generation is what? 16. In the F2, in the monohybrid, what is the total progeny in the F2 generation? 4 only. How many boxes you are seen in the Punnett square? 4 boxes. In the dihybrid, how many boxes are there? 4 into 4. How many boxes are there? 16. This is the total progeny obtained in the F2 generation. So just see, phenotypic ratio is 9:3:3:1, and the genotypic ratio 2:1, 2:1, 2:1, 4, 2:1. Ratio you can write anyway. Ratio never changes. 4 2s, 4 1s, you can remember blindly. Is it clear?
Okay, sir.
Okay. Next slide. You can go for. What happened? somewhat different. Now, sir, it is visible?
Yes, sir, visible.
Okay, okay. Now, dihybrid cross. I will remove this diagram. Okay. Next, trihybrid. Same in the way. In the trihybrid cross, it involves three contrasting traits. At a time, we can consider three contrasting traits observed in the parental plants, and which represented by three alleles. Here, three alleles are there. So when we are getting F1, all are dominant, or capital Y, capital I. So these three respective characters are expressed in the F1 plants. Remaining characters are missing. And we obtain this ratio 27:9:9:9, 3:3:3:1 phenotypic ratio. So it confirms the segregation of multiple traits. So we can explain in the next slide what is the type of inheritance laws are expressed. So why Mendel was successful during his hybridization experiments? First of all, used pure lines and controlled crosses, studied discrete and contrasting traits, applied mathematics and probability, large sample sizes for statistical accuracy, maintained clear and accurate records. And every time he noticed that what progeny reveals the parental characters. How many parental characters are seen in F1 generation, in F2 generation? So based on these results noted correctly and maintain the records for several generations. Based on the all generation values, he summarized the values in a mathematical ratio. Based on these mathematical ratios, he explained the inheritance laws. So, and test cross and back cross also, very well. This is one of the important aspect in many competitive examinations and PG entrance examinations. Test cross and back cross. So, test cross, a cross made between F1 hybrid with recessive parent. We obtain a what type of phenotypic ratio and what type of genotypic ratio go through the Punnett square box. And when we can refer back cross, the same F1 hybrid plant cross made with either any parental plant, maybe whether it is a homozygous dominant or homozygous recessive. So what is the main purpose of this test cross? So for example, I select two parental plants. One of the parental plant shows tall character. The tall character in the homozygous or heterozygous. You can test the parental plant in heterozygous condition or homozygous condition. How can you know whether it is homozygous or heterozygous? So that's why we are doing test cross. If it is test cross result is same, we can confine that the parental plant in heterozygous condition or homozygous condition. So I will check this by this table. You can easily understand that one. So this is the test cross and back cross related interpretation. A graphical representation of the test cross. So for example, this is the one of the parental plant. Okay. So one of the parental plant. That is what dominant phenotype. So the color of the flower we can see here. So color purple is there. White is the recessive. So once I will check this plant having this type of genotype or this type of genotype, whether it is in the homozygous condition or heterozygous condition. Hello? అర్థమవుతుందమ్మా? Hello? Anybody is there?
Sir, can you repeat again?
you select two parental plants. One of the parental plant shows dominant character. Yes, but the dominant character representative genes in homozygous condition or heterozygous condition. I want check to test the tall character or color of the flower in dominant color in heterozygous condition or homozygous condition. If it is homozygous, is nothing but capital. If it is heterozygous, is nothing but is called what? Capital smaller. Once it is crossed with homozygous recessive, once it is crossed with homozygous recessive. So what kind of result achieved? So based on the result, I will explain that dominant character plant may be homozygous condition or heterozygous condition. See here, for example, we assume that capital cross with small r. So what we are expecting here, all flowers are violet. All flowers are violet because from one side the gamete receive small letter, and another side gamete receive only capital R. So capital R and small r whenever combined, we are getting dominant character only. So that's why all flowers are violet. The unknown flower is homozygous dominant. ఓకే, అంటే ఓన్లీ వన్ కైండ్ ఆఫ్ ప్రోజనీ వచ్చింది ఇక్కడ. సో ఇటువైపు కనుక ఇలా ఉంటే ఎలా వస్తుంది చూడండి, క్యాపిటల్ ఆర్ స్మాల్ ఆర్, వన్ గ్యామెట్ రిసీవ్డ్ క్యాపిటల్ ఆర్, అనదర్ గ్యామెట్ రిసీవ్డ్ స్మాల్ ఆర్. సో వన్స్ స్మాల్ ఆర్ గ్యామెట్ fertilized with a small r gamete, we are getting what? Recessive color flower. So half of the flowers are violet and half of the flowers are white. So it indicates what? Our test, our parental plant is in which condition? Heterozygous condition. So the test cross result is 1:1. Definitely our parental plant in which condition? Hello? Hello?
Sir.
Our test cross results shows 1:1, either phenotypical or genotypical ratio. So that indicates what? Our parental plant in which condition?
Heterozygous.
Yeah, heterozygous condition. If we get all progeny with only one trait, one character, our parental plant character gene is in dominant genotype, dominant homozygous type, dominant homo. That's why we are using test cross. Test cross ఎందుకు వాడతారు? పేరెంటల్ ప్లాంట్ లో ఉన్నటువంటి క్యారెక్టర్ సంబంధించిన జన్యువులు విషమయుగ్మజంగ స్థితిలో ఉన్నాయా, సమయుగ్మ స్థితిలో ఉన్నాయా తెలుసుకోవడం కోసం మనం టెస్ట్ క్రాస్ చేస్తాం. కాబట్టి టెస్ట్ క్రాస్ యొక్క ప్రధానమైన ఉద్దేశం ఏంటంటే పేరెంటల్ ప్లాంట్ మనకి ఎప్పుడు ప్యూర్ హోమోజైగస్ గా ఉండాలి. కాబట్టి మనం వాడే పేరెంటల్ ప్లాంట్ హోమోజైగసా హెట్రోజైగసా అని తెలియాలంటే టెస్ట్ క్రాస్ చేయాలన్నమాట. ఐ థింక్ అండర్స్టూడ్ ఎవ్రీవన్. అర్థమైందా?
Clear, sir.
Clear. Okay, very nice. So whenever got the test cross result is 1:1. If it is dihybrid, 1:1, 1:1. Is it clear?
Yes, yes.
Okay. So next we can go for another important one.